Okay. Alrighty. Okay, this time we're going to continue talking about plants and water. And we're going to talk about I would say, the most difficult concept in plant physiology, which is water potential. So hopefully I've set the stage that water tends to move passively in plants, but also in other places. And there are basically two driving forces that make water move. One is there's concentration differences of water. Water moves down its concentration gradient like any other molecule, diffusion. And then there's pressure. And you know, in plants, there is positive pressure in cells. You can imagine this pushes that water out of the cell. And in other places in the plant, there's actually negative pressure or tension that sucks water through the xylem. So we have these two forces, and together they form water potential. So today we're going to have a look at that. So this was the last slide. You saw the concept of water moving down its concentration gradients. And if you look at it from the other side, from the side of the solutes, which are dissolved in water, you can say water always wants to go to where the solute concentration is highest, because that's where water concentration is lowest. So wherever you have a lot of solutes, dissolved particles, that's where water will want to go. And there's a term. If you have two water solutions with a different concentration, then we call the one with the higher concentration hypertonic relative to the other one, and the one with the lower concentration of solutes, we call it hypotonic relative to the other one. So the water will move from the hypotonic side of a membrane to the hypertonic side, where the most solutes are. If the concentration is the same, then we say that it's isotonic. Those are some concepts that you saw last time. So as an example, if you have a cell with a certain concentration, let's say 0.3 molar, this is totally an arbitrary number, and you throw it in three different situations. You throw it in a very high solute concentration, like seawater, then the water will want to go the water will want to go out of the cell into that seawater because the concentration of solutes is higher in the seawater. So that cell is going to shrivel up. That's called plasmolysis. This is going to kill a cell. So if possible, an organism will try to avoid that. So if it's thrown in a situation with a very high solute concentration, the best it can do is to try to increase its own solute concentration, maybe by breaking down larger molecules into smaller ones. Because when we're counting solutes, any solute is equal. Whether they're small or big, doesn't matter. So if you have a sucrose and you break it down into a glucose and fructose, suddenly you have double the solute concentration without doing much except for snipping that bond. So that's a way in which a cell can respond to an environment that suddenly has more solutes and it's threatening to lose water. It can increase its solute concentration. But sometimes it cannot be avoided, and then you may get this shriveling up and dying. You can also have a cell being in an isotonic environment. This is actually required for animal cells. They have no cell walls that can protect them. They have to be exactly right. But plant cells, they're okay with it for a little while to be in an isotonic environment, but not in the long run, because cells want to have positive turgor in order for them to have their stomata open and have good gas exchange and keep taking up that CO2 and growing. So if you look at a plant in this condition, it will look wilted. It's not really happy. It will survive for a while, but not in the long run. What plants want to be is in a hypotonic environment, like water, that's fine, or fresh water. And then what's going to happen is water will want to move into the cells. That means the vacuole is going to blow up. It's going to have lots of turgor. And then the plant is happy and fresh looking, crispy. And then it can open its stomata. It can have gas exchange. It can grow. So that's just a concept of how when a plant cell is happy, it wants to be it wants to have a higher solute concentration in its environment. So now we're going to have a look at the pressure component. And if we go back to a simple example, you saw this kind of situation before where you had a simple glass tube filled with a solution, and it is in contact with another solution, in this case water. So there are solutes within this glass tube there's a solution there. And on the outside of the glass tube, there's just water. And in between, there's a membrane. And the solutes cannot pass through this membrane, but the water molecules can. So the water molecules are going to move toward the solution. That's what you see with these arrows, because water will move down its concentration gradient. And the water concentration is lower in the tube because there are solutes there. So the water concentration is lower. At some point, the water level is going to rise and rise and rise, and at some point it stops. And that's probably because pressure is going to build up. Gravity is going to pull down on that water column. It's going to push on this membrane. So we already have here this tug of war starting to happen. The solutes are dragging up the water into the solution, but the pressure is going to build up from gravity and it's going to push back down. At some point, we'll have equilibrium. So those two aspects. Now, what if you would take a plunger and you would push down, you would force down that water back down? Then what you would have to do is you would have to put some pressure on there, because the water will naturally tend to want to move in up to this point, but you don't allow it to do so. You have a plunger there. That will create a positive pressure. And you could actually measure this if you had a little gauge, a little measuring device. You could actually measure this. And you can imagine that the more solutes you have, the higher the concentration in this glass tube, the more pressure you would have to put on the system to prevent that plunger from being pushed up. So that would be osmotic pressure or osmotic potential, which is kind of the same thing. So the osmotic potential is written here as this PSI, this Greek letter, PSI or psi. Psi S, S for solute. So the osmotic potential, or also called solute potential, is the potential of solutes in a solution to attract water through a membrane via osmosis. The more solutes there are in a solution, the more negative the solute potential is. So it's expressed as units of pressure. So it's kind of a suction potential. It has the potential to suck water towards it. Those solutes have that potential to suck water towards them. This is a negative pressure, a suction. Like if you suck on a straw, you create a negative pressure, a tension, and that's going to suck up the water. So it's kind of a similar thing. Solutes have the same suction effect. So the more solutes you have, the higher the solute concentration, the more negative this osmotic potential or solute potential. If you would have pure water with no solutes, then the solute potential is 0. That's how it's defined. And as soon as you have even one solute, the solute potential gets a little bit negative, and the more solutes you have, the more negative it gets. Within a cell, you often have a solute potential of about 1 megapascal. So that is a pressure unit that's typically used. And again, it's the number of dissolved particles that counts. You can add them all up. It doesn't matter how big they are. As long as they are dissolved, they contribute to the osmotic potential. So a cell can regulate its own osmotic potential by breaking down solutes into more smaller ones, or it could also do the other thing. It could combine them into fewer bigger ones. But that way, cells can regulate their osmotic potential in response to its environment. If the environment gets drier, then the plant will need more solutes so it can lower its osmotic potential, but make it more negative by creating more particles, by cutting bigger ones into more smaller ones, because each particle counts equally. So I just said megapascal. What is a megapascal? A megapascal is 10 to the 6th Pascal. And actually, maybe you're more familiar with just atmospheric pressure around us, and we often also talk about that as bar. Atmospheric pressure is defined as 1 bar, and that corresponds to 100,000 pascals, so 10 to the 5th Pascal, or 0.1 megapascal. That is atmospheric pressure, just to give you an idea. So if you have, like I just said, a typical plant cell may have a solute potential of minus 1 megapascal. It would be 10 times higher than the atmospheric pressure, but it's not really air pressure like we're used to, but it's kind of water pressure. Okay, so pure water is defined as having a solute potential of 0. It does not have the capacity of solutes to attract water toward them. They have no solutes. It's pure water. But as soon as you have solutes in there, then it becomes negative. Solute potential is never positive, okay? It's always 0 if it's pure water, or it's negative. And the more solutes, the more negative. So to give you an idea, in a cell, I already said it's about minus 1 megapascal, but it can vary because it depends on the species, and also it depends on the conditions. So like I said, let's say typically it's -1, but if a plant has just been drought stressed, it may actually be -1.5 or even -2. And that could either be because the plant has lost water through evaporation, simply concentrating the cell fluids, but then the cell would not be happy at all. You would see it looking very miserable. But more likely, if you see that the solute potential is becoming more negative under drought conditions, the plant is on purpose lowering its osmotic potential by breaking down larger molecules into smaller ones. That's called osmotic adjustment. So, but also if you have a desert plant, just a random desert plant, if you would test it, even if you give it water, make it as happy as you can, then still it will probably have a lower osmotic potential than a plant that normally lives under very like tropical, full of water conditions. Just they are adapted to have a high solute concentration. That's just normal for them because in nature they always have dry conditions. So it's kind of standard, always have a lot of solutes. So it depends on the species, what they're adapted to, how many solutes they have. And then also it can respond to the environment. So that gives you a little bit of idea that solutes can have the capacity, the power to attract water, and we can express this in terms of solute potential, which is a negative number. So that's one factor that drives water across membranes. Now the other factor I already said is pressure difference. And you saw in the example of this osmometer, as it's called, this glass contraption, that if you have a system that is now closed because you have a plunger on top of it, then the solutes in the system want to attract water because of their solute potential. But what you get is the water will move in to some point, but then you have a buildup of positive pressure. And this is what in cells you would call this turgor pressure. But in a system like this, it would just be pressure because it's not really a living cell. And this is another component, another driving force for water through a membrane. It's the pressure difference. So there's the solute difference on both sides of the membrane, and then there's the pressure difference across the membrane. So in this system, we have the solutes kind of sucking the water molecules through that membrane. So that's inward, and then we have the pressure building up, pushing the water molecules back out. Two driving forces that go in opposite directions. So the term for this component of osmosis is pressure potential. So what is osmosis? It's the sum of these two forces. So it's the solute effect and the pressure effect. They are going to both affect the passive water movement through a membrane. So the process we call osmosis, water movement through a membrane passively, but that osmosis is controlled by these two main forces, solute difference and pressure difference. So the pressure potential, or psi p, is the other main component that drives osmosis, pressure difference across the membrane. And if you have a system that is open to the atmosphere, then it's defined that the pressure potential, psi p, is 0. So in reality, it's 1 bar or 10 to the 5th Pascal, but it's defined as the pressure potential is 0 when there's just atmospheric pressure. If you have a situation like here on the right, where you have a closed container, a pressure has built up, it's bigger pressure than atmospheric pressure. Now psi p is bigger than 0. Similarly, in cells, of plant cells, the psi p is bigger than 0. There's turgor. But if you would have a wilted cell, a flaccid cell, then psi p is 0. It's just it's not bigger than the environment, it's also not lower than the environment. It's 0. But in the xylem, psi p is actually negative because there's suction going on there. There's transpiration going on from the shoot, and that's sucking up water through the xylem. So within a plant, in the xylem, you can have a negative turgor pressure. Psi p is smaller than 0. In a flaccid cell that is wilted, normally in plants you don't really have that, but you can have it in a system, that if you just manipulate a cell, you can get the cell to be flaccid, then psi p would be 0. And in a happy cell, psi p is positive. And it can actually be quite positive, so comparable to a car tire or a bike tire. It can be quite high. So now let's combine the two. So we have these two components that drive osmosis. These two main components, we have the solute difference and we have the pressure difference. So to what extent is the water going to move? Until we have equilibrium. So the forces are equal that move it in one direction and that move the water in the other direction. Let's say solutes drive it into the cell, pressure pushes it back out of the cell. These two forces cancel each other out at some point when we have equilibrium. So how do we express those two forces in one go? That's why people came up with the term water potential. So it's the sum of those forces, the sum of the solutes effect and the sum of the pressure force, combine them, and then we call it water potential. And water potential is shown as this Greek letter psi, psi. And psi comes from the Greek god of the sea, Poseidon. So psi, psi, this is his trident, you see, that he's holding. So water potential psi is also sometimes written as psi w with a little subscript w. That is the overall potential of a solution or a place to attract water via osmosis. And there are these two main driving forces, the pressure and the solutes. If you add them up, so the sum of those two, then you get the overall water potential. And we'll see some examples to make it clearer. But just to reiterate, we have a certain place in a plant or anywhere else, it can also be a solution, has a certain solute concentration. If there are no solutes, if it's pure water, then psi s is 0. But as soon as you have some solutes, psi s becomes negative. So that's the one component. And then the other component, pressure. If you have an open system, open to the air, then psi p is 0. But if you have in a plant a positive pressure, like inside a cell, psi p is going to be positive, and in the xylem, psi p is negative. And then if you add those up, so you can be in a root cell, for instance, you have a certain concentration of solutes that gives that root cell a certain psi s. Within that root cell, you have turgor that gives that root cell a certain psi p. If you add those two up, then you get the overall water potential. So you get psi or psi w. If you now want to know, is this root cell able to take up water from the soil? Or is that root cell going to be able to deliver water to the xylem? Then what you can do is you take that overall water potential and then you compare that with the overall water potential of the soil and with the overall water potential of the root xylem. And then you will know whether the water is going to move into that root out of the soil or not, and whether that water is going to move from the root to the root xylem for uptake to the shoot or not. So just with one easy number, the overall water potential, you can now know, is the water going to move or not? It combines those two forces of solute differences and pressure differences. And the rule is, water will always move to where the overall water potential, psi or psi w, is the most negative. That is the universal rule, no exception. Water will always go to where the overall water potential is the most negative. So if a root wants to be able to take up water from the soil, its water potential must be more negative than that of the soil. No matter what the solute potential or the pressure potential are, as long as the overall water potential is more negative, water is going to move into that root. So this is the one thing that's always true. So if water is going to move into roots, for instance, from the soil, then obviously the water potential must be more negative in that root. Why would water potential be more negative in the root than in the soil? What's going to be the overruling force? It's not the turgor. Turgor actually is positive inside those root cells. Turgor is trying to push the water out of the root into the soil, but still the water is going to move into that root. Why? Because the solute concentration is so high. So the solutes win, you could say. The solutes win over the pressure. And then we say it's solute-driven. So if we have water movement into the root from the soil, then it's osmosis because it's water movement across a membrane. That's always osmosis. And when it moves, it can be driven by the solutes. They kind of overrule any pressure differences there are. And they're going to move it across the membrane, in this case, into the root. There's enough solutes in that root to overrule the pressure differences, and it's going to go into that root. But on the other hand, if, for instance, root cells are going to lose water to the xylem, then actually the solute concentration is higher in the root cells than in the xylem. The xylem is apoplast, and the solute concentration is always lower outside of cells than inside cells. So it's less negative in the xylem. The solute potential is less negative in the xylem than in the root cells. Still, the water is going to move from the root into the xylem, from the root cell into the root xylem, because the pressure difference overrules, in this case, is more important, is bigger than the solute difference. Because the xylem has all this negative pressure because of suction going on up in the shoot, which is sucking like on a straw on that xylem. So now it's the pressure that wins over the solute concentration. So then we say it's turgor-driven. So it's still osmosis. Water moving from root cells into root xylem is osmosis because it's water movement across the membrane. And in that case, it's turgor-driven. So the turgor overrules the pressure difference, the solute concentration difference, and it pushes that water out of the root cells, and then it becomes available for transpiration up in the shoot. So those are the two main forces in water potential that drive water movements in general in biological systems. We have pressure differences across membranes. We have solute differences across membranes. And both of them contribute, pushing and pulling on the water, to the overall water potential psi. In some systems, there are additional components. For instance, if you have very dry situations like a seed, there's hardly any water in seeds, and also in soils, which tend to be very dry, then you have what's called the matrix potential, or psi-m. And this is the potential of non-dissolved molecules to also bind water. As an example, you probably know if you have cotton clothing on and you're in the rain, they get very wet. They hold on to the water very much. Still, cotton is insoluble. It's cellulose. It's solvable. Why does cotton bind water? Because it's very hydrophilic. It binds the water. It doesn't dissolve into it, but it binds it. Similarly, soil particles tend to bind water just by hydrogen bonds. So that is the matrix potential. Usually in normal plants, like crops in the field, whatever, plants in the greenhouse, matrix potential is negligible. So we say, forget about it. It's so small it doesn't matter. But if you're dealing with seeds, or if you're very interested in soil properties, plant-soil interactions, matrix potential can be a factor. And then another component that usually is not a factor only in trees is the gravitational potential, or psi g. This kicks in above 10 meter height, and then it goes up with 0.1 megapascal for every 10 meter. So if you have a 20 meter high tree, so that would be about 66 feet, then it would be 0.2 megapascal. If you have a 100 meter high redwood, it could be as much as 1 megapascal. So that it adds a positive 0.1 megapascal for every 10 meter. And like I just said, matrix potential, that contributes a small negative component, so it kind of sucks the water toward it, so it's a negative force. So that is some other components that in practice you will not have to deal with much. But I hope you get these two components, the effect of pressure, pushing water across membranes or sucking water through membranes, can go either way. And solute potential, that is the effect of solutes dragging water towards them through a membrane. So those together are the main driving forces for water movement across membranes, osmosis. So we sum those both up and we call it water potential. And wherever that water potential, that sum of those two components is the most negative, that's where the water will go, no exception. Any questions about this very difficult concept? Who would say, yeah, that's pretty clear? Okay. Who would say, well, you totally lost me? Okay. Okay. So I hope if I lost you that it will become clear when we start looking at all these examples, first at the cell level and then at the whole plant level. And we'll recap it, we'll do some practice with it, and then hopefully it will become clear, and you're always welcome to come talk to me if it's not clear. So here's an example. We have a flaccid cell, and we throw it in a sucrose solution that has a concentration of 0.4 molar. So first there is a formula which I'm going to show you later, by which means you can actually calculate, if you have a certain solution, what the solute potential of that solution is. But for now, just accept it as a given that a 0.4 molar sucrose solution has a solute potential of minus 0.9 megapascal. So this solution has a water potential. So water potential is not something that only biological systems have, but also just a body of water or soil. So non-living systems also have a water potential. So in this case, this sucrose solution has a water potential. The solute potential is minus 0.9. It's defined by the sucrose in there. The pressure potential is 0 because it's open to the atmosphere. And the water potential, psi, is the sum of the two, so minus 0.9. So now if we look at our cell, it has an overall water potential of minus 0.7 megapascal. That is just, and we know it's flaccid, so that means that there's no turgor. So we know the pressure potential must be 0. And then you know automatically that the solute potential must be minus 0.7 because it's the sum of pressure potential and solute potential is water potential. So if you know 2 of the 3, you can calculate the third. So in this case, you may be told a flaccid cell has a water potential of minus 0.7 megapascal. Then you know, aha, flaccid, pressure 0. Water potential equals pressure potential, 0, plus osmotic potential. Therefore, osmotic potential must be minus 0.7. So that you know, now you have a solution with a water potential of minus 0.9. You have a cell of water potential minus 0.7. You throw the cell in the sucrose. What's going to happen? Well, you just heard water potential. Wherever water potential is most negative is where water will always go. So where is it most negative? Sucrose. So the water is going to leave the cell. The cell is not happy, but it can't do anything about it. So it's going to lose water. It's going to shrivel up. It's probably going to die. At some point, there's going to be equilibrium. So what's going to happen to the pressure? It's an open system, so pressure is not going to change. It's 0. It stays 0. There's no vacuum created, so pressure is going to stay 0. Water potential of the cell is going to change. It's going to become the same as that of the solution, so minus 0.9. So that means, since pressure is still 0 and the water potential is minus 0.9, that the solute potential must have become minus 0.9. And that makes sense because water has left the cell, so the existing solutes are now more concentrated. The concentration is higher. So that's why the solute potential is more negative. So yeah, the question is this a situation where a cell could actually respond by breaking down some solutes and lowering its osmotic potential? Yes, this cell would love to try to do that if it can do that in time. If you throw one cell in a solution, it will probably not have the time. But if it goes gradually, like in nature, then it could be that the cell, the plant as a whole, can be there and change its metabolism in time to avoid this happening. So this is one situation. Now we have another situation. We have exactly the same cell. It's flaccid. It has a water potential of minus 0.7 and therefore a solute potential of minus 0.7 because pressure potential is 0. It's flaccid. Now we throw it in distilled water. What's going to happen? Who is most negative in terms of overall water potential? The cell, minus 0.7. So water is going to move into that cell. Does that cell like that? Yes. It will not burst because it has a cell wall. So the water is going to move into that cell, and we're going to have a buildup of pressure. And now there's something going to happen that I think is a little counterintuitive, but even though water moves into that cell, the solute concentration is not going to change significantly. It may change a little bit, but so little that it doesn't significantly change from minus 0.7. And that is because the cell wall is so rigid that you have a lot of pressure buildup before the cell volume can actually change significantly. It's like having a wooden box and trying to push more water into that wooden box. You only have to add a little bit of water to get a big pressure buildup because there's no space for that water to go. So what's going to happen effectively is, at equilibrium, the water potential of the cell is going to equal that of the solution. So the cell is going to end up with a water potential of 0 megapascal. The solute potential is not going to change significantly, even though some water has come in, but it's so little that it doesn't, maybe it's now minus 6995 or something, but it's still effectively minus 0.7. So how can the water potential be 0? Because now we have a pressure potential of plus 0.7 at equilibrium. So the overall water potential is the same as that of the water around the cell. And remember that 0.1 would be atmospheric pressure, so this is 7 times higher than atmospheric pressure, and that's way higher than even a bike tire, which may have like 3 or 4 bar. So this is twice as high. So plant cells can withstand a lot of positive pressure because of that woody cell wall that they have. So this is actually something that cells would like to promote. They want to build up that positive pressure. So effectively what happens when you throw a cell into a solution, there's going to be water movement. Unless they start out at the same water potential, there's going to be water movement. The water is going to move to whichever side of the membrane the water potential is most negative. So in one case you saw it was the solution. In the other case it was the cell. The water is going to keep moving until we have equilibrium, where water potential is equal on both sides of the membrane. So who has changed to make that happen, to reach that equilibrium? The cell. The solution doesn't change. Even if water would move out of a cell, the solution wouldn't change significantly because the cell is infinitely smaller than the solution. So forget about the solution changing. The solution doesn't change, the cell changes. Okay, so then what can change in the cell? It depends. If you have a cell that is already plasmolyzed, then the pressure is not going to get any lower than 0. And in that case, the solute concentration can actually change when the cell is totally going to shrivel up. But in more natural systems, you typically have that the pressure changes when water moves into cells. The pressure quickly builds up without significantly affecting the solute concentration in the cell. So the solute potential in the cell stays the same. Effectively, pressure builds up, and that makes it possible for the cell to reach the same water potential as the solution. So here we have another example, a third example, which is the most complex. We have our flaccid cell again. In this case, it has a water potential of minus 0.732. It's flaccid, so no pressure potential. Solute potential, therefore, is the same as the water potential. Now we throw it in a solution of 0.1 molar sucrose. So we throw it in. What's going to happen? Who is most negative? The cell. Cell is minus 0.7. And the 0.1 molar sucrose actually has a, where's the point? Oh, here's the 0.1 molar sucrose. It has a solute potential of minus 0.244. And a water potential of minus 0.244 because it's open to the air, so its pressure potential is 0. So our sucrose has minus 0.244, our cell has minus 0.732. Water is going to move into the cell. It's more negative until we have equilibrium. At equilibrium, the cell has adjusted. The solution doesn't change. The cell has adjusted. It is now the same as the solution. Its overall water potential is minus 0.244. Its solute potential has not changed significantly, even though a little bit of water has moved in. But even with that little bit of water, it has already built up so much turgor, so much pressure, that the cell has now reached equilibrium with its environment. So now the question can be, at equilibrium, what is the water potential, the solute potential, and the pressure potential of our cell? That could be an exam question, for instance. So at equilibrium, the cell has adjusted. The water potential of the cell has become the same as the one of the solution. The solution hasn't changed. So how has this cell adjusted? The solute potential is still the same because although a little bit of water has moved in, that hasn't significantly changed the solute concentration. However, the pressure has built up tremendously. So how high is that pressure? The overall water potential at equilibrium of the cell must be minus 0.244. That's the same as the solution. Solute potential has remained the same. It was minus 0.732. That was given. It still is the same. So now you can make the simple equation that the pressure potential, the turgor, must be the water potential minus the solute potential. That's the only equation, incidentally, that you need to know for the exam is water potential equals solute potential plus pressure potential. It's a very simple equation. So if you know the water potential and you know the solute potential, you can calculate the pressure potential because that must be the water potential minus the solute potential. So, and then you have to be careful with your signs. So water potential is minus 0.244. And you want to do that minus the solute potential, which is also negative. So you say minus 0.244 minus 0.732, which effectively is a plus, right? Two minuses make a plus. So minus 0.244 plus 0.732, and then you end up with 0.488. And then use your common sense. Does that make sense? That the pressure potential is plus 0.488? Yes, because we have turgor now. If you would end up with a negative pressure, recheck your calculation. You must have made a mistake. In a cell, you will not have a negative pressure. In a xylem, you could have, but not in a cell. So that's how you do these kinds of. I hope you can kind of see the driving forces of osmosis and how this has implications for whether water will move into cells or not, and how that can be the difference between life and death of a plant. And how plants can actually influence this situation by increasing their solute concentration. Any new questions now? I hope these examples made things a little easier to follow. Okay. And I just said, just accept for now that if you know the concentration, then you know the water potential for certain chemicals. And this is actually that equation. You don't have to memorize it, but I do want you to understand it. So this is how you can calculate solute potential when you know which molecule is in a solution and what the concentration is. So it's the formula is minus MERT, where don't forget the minus. M is the molality, which is kind of the same as the molarity. It's just mole per kilogram rather than mole per liter. Effectively, it's the same because a liter of water is also a kilogram of water. If you would have other solutes, then it might be a little bit different. So this is effectively the molarity, so the number of particles per volume of water. Then you have I here, which is the dissociation factor. It's the number of particles that one molecule of this compound dissociates into when it's dissolved in water. So if something doesn't dissolve at all, like starch, I would be 0. It doesn't dissolve at all. If you have something like glucose, if you throw a glucose molecule in water, it stays glucose, it doesn't fall apart or anything, then I would be 1. If you have NaCl, which falls apart into two pieces, Na plus and Cl minus, I would be 2. If you have magnesium chloride, it falls apart into three pieces, Mg2 plus, Cl minus, and another Cl minus, I would be 3. So that's why I is in this formula. So the more pieces it falls into, the bigger I is. R is always the same. It's the gas constant, which is 0.00831. And T is the absolute temperature in Kelvin, which follows the same. It's kind of like Celsius, except that the 0 point is at minus 273 Celsius. So 0 Celsius would be plus 273 Kelvin. And 20 degrees Celsius would be plus 293 Kelvin. So 1 degree difference is the same for Kelvin and Celsius, but it just starts where 0 is for Celsius is 273 for Kelvin. So example, if you have 0.1 molar of sucrose solution at 20 degrees Celsius, then the solute potential of that solution would be minus, don't forget the minus, and then 0.1, that's the molarity, times 1, because sucrose does not fall apart into pieces. And then you get R, and then you get the temperature. 20 degrees Celsius is 293 Kelvin. And if you then make that calculation, you get minus 0.24 megapascal, which is what you saw earlier in that example question. Okay? Does it make sense? So what this shows is that the higher the molarity, the more negative the solute potential, which is what you already knew, but now you see it in a formula. You also see that the temperature can have an effect on it. So the higher the temperature, the more negative at the same concentration. And you see clearly that the degree to which a molecule dissociates influences, or dissolves also, influences its contribution to the solute potential. So that is for a solution, how to calculate the water potential of a solution. Air can also actually have a water potential. Soil can have it, water can have it, and air can have a water potential. And that also has a formula. It's this one. The water potential of air is 1.06 times, here we have that absolute temperature again, times the log of the relative humidity. So relative humidity of 50%, for instance, then RH would be 50 there. And here in Colorado, we may have only as little as 10% relative humidity. Then where it says RH here, you would put 10. So if you would do a calculation, and you would say, okay, let's calculate for a 50% relative humidity air what the water potential is, then you would come to an astonishing minus 0.93 megapascal. Isn't that amazing? That's 50% relative humidity. How negative that water potential is, let alone Colorado. Just imagine how negative that is. And now imagine your plant. And your plant has a water potential of minus 1 or something. What if it opens its stomata? That water is going to rush out of those stomata into the air. So that's why why plants have to be so careful opening their stomata, because they're going to lose water like crazy when they do. So it's just good to see these equations and to just to realize how kind of threateningly dry the air is for plants. The last couple of slides, I'm going to tell you about how you can measure water potential, and also just some examples of pressure. Here we see here, for instance, a car tire is typically inflated to about 0.2 megapascal. The water pressure in your home, when you open your tap, is also about 0.2 megapascal. If you're underwater, maybe you've swam underwater and you felt the pain in your ear, if you're at 3 meters deep, 10 feet deep, that's only 0.05 megapascal, and that's at 5 meters deep. And in a cell, it can be as much as often 3, 5, or 7 megapascal turgor pressure, just to realize, wow, this is really high pressure in these plant cells. And here to give you an idea of the solute potential in cells, it's typically maybe around 1 megapascal. If you compare that with seawater, seawater is about minus 2.6. So cells are about 40% as salty as seawater. But of course, the solutes are not just salts. In plant cells, there are also sugars and acids, anything, minerals. And this also shows you the effect of temperature. It becomes a little bit more negative with the higher temperature. So that's just good to realize. And up here you also see some other pressure units, because I'm talking megapascal all the time. I already mentioned 10 to the 5th megapascal is 1 bar. If you're more used to thinking in pounds per square inch, you can see the comparative number here. 14.7 is equal to atmospheric pressure. 10 to the 5th Pascal. Okay, so how do you measure pressure, water potential? One thing you can use is a pressure bomb, as it's called, which sounds very dangerous. If you're traveling on an airplane with a pressure bomb, you probably don't want to mention this. I have a pressure measuring device. So the pressure bomb, what it does is you put a whole plant, you can cut a whole plant or at least a branch, and you stick it in this chamber with the end sticking out. And this is then sealed off very tightly. And now you start to put pressure with this air cylinder on the system, on the plant, and you wait for the droplet to come out, to be pushed out. And then you measure on this pressure gauge what the pressure is at that point, and that is equal to the water potential of this plant. So if this plant had a more negative water potential, maybe it's a desert plant, you have to put more pressure on that plant to push that water out. So that's one way to do it. This is a very fast way. Just in minutes, less than a minute, you can get a reading. So that's an advantage of this system. But you have to kill the plant for it, or at least destructively sample it. That's a disadvantage compared to other systems. Another system is this one. It's called a psychrometer. And it's actually quite a small chamber. You can just hold it in the palm of your hand. It looks really big here. And you can unscrew it and screw it back on. It has this little chamber inside, very small. And what you do is you take a leaf disc with a hole puncher. You can just use any regular paper hole puncher. So the size is from the hole in the paper that you have on your desk. And you just carefully put that in a little holder. You stick it in this little chamber. You screw it closed so it's sealed off. And then you wait. And then you have to wait typically at least 10 minutes, maybe as much as 45 minutes. You wait till you have equilibrium. And then at that point, what you do is you measure the relative humidity of the air in the chamber, and that's going to be in equilibrium, so it's going to be the same. The water potential in that air is going to be the same as that in the leaf. So by measuring the relative humidity, you know the water potential of the air, and then you know the water potential of the leaf disc, because there was equilibrium at that point. So there's a little machine that you can carry, and you take the psychrometer, you plug it into the machine, you let it equilibrate, and then you measure. There's a little thermocouple in here that you can cool and then or heat and cool, and then the condensation onto that thermocouple is going to tell you the relative humidity of the air. So that's called a psychrometer, and this is called a pressure bomb. Okay, that's all for today.